Y-Chromosomes Traveling South, the Cohen modal haplotype and the origins of the Lemba, the Black Jews of Southern Africa. The Lemba are a traditionally endogamous group speaking a variety of Bantu languages who live in a number of locations in southern Africa. They claim descent from Jews who came to Africa from Sinai. Sinai is variously identified by them as Sana'a in Yemen, Judea, Egypt, or Ethiopia. A previous study using Y-chromosome markers suggested both a Bantu and a Semitic contribution to the Lemba gene pool, a suggestion that is not inconsistent with Lemba oral tradition. To provide a more detailed picture of the Lemba paternal genetic heritage, we analyzed 399 Y chromosomes for 6 microsatellites and 6 biallelic markers in 6 populations: Lemba, Bantu, Yemeni Hadramaut, Yemeni Sena, Sephardic Jews, and Ashkenazic Jews. The high resolution afforded by the markers shows that Lemba Y chromosomes are clearly divided into Semitic and Bantu clades. Interestingly, one of the Lemba clans carries, at a very high frequency, a particular Y chromosome type termed the Cohen modal haplotype, which is known to be characteristic of the paternally inherited Jewish priesthood and is thought, more generally, to be a potential signature haplotype of Judaic origin. The Bantu Y chromosome samples are predominantly greater than 80% YAP+ and include a modal haplotype at high frequency. Assuming a rapid expansion of the Eastern Bantu, we used variation in microsatellite alleles in YAP+ Psi81G Bantu Y chromosomes to calculate a rough date, 3,000 to 5,000 years before the present, for the start of their expansion. Introduction. The Lemba, once referred to as Kruger's Jews because President Paul Kruger, president of Transvaal during 1883 to 1900, was thought to have discovered them, are commonly referred to as the Black Jews of South Africa. Their claim of Jewish origin is based on slim evidence, a persistent oral tradition of uncertain antiquity and a number of suggestive customs, from circumcision to food taboos, which appear to be Judaic, but could be Muslim or, indeed, in the case of circumcision, African. Mandavenga 1983. Lemba tradition holds that the tribe came from Sinai in the north by boat. The original group is said to have been entirely male, with half of their number having been lost at sea. The remainder made their way to the coasts of Africa. Once there, they rebuilt their city of Sinai, later leaving it to build a second city of the same name. Sinai is variously identified by the Lemba as Sana'a in Yemen, Judea, Egypt, or Ethiopia. Ruwita 1997, Parfit 1997. The first clear and unambiguous reference to the Lemba as a separate tribe and perhaps polity is from a Dutch report from 1721, Lezenbang 1977. Today the religious life of the Lemba is highly syncretistic. Many of them belong to various Christian churches, e.g., the Zion Christian Church and Pentecostal groups, whereas some in Zimbabwe are Muslims. Others, however, claim to be Lemba by religious practice as well as by ethnic identification. The religious practices of these Lemba do not have much in common with Judaism as it is practiced elsewhere. There are thought to be 50,000 Lemba spread over South Africa and Zimbabwe, with some closely connected groups in Malawi. Parfit 1997. At some time in the past they became scattered among the more powerful neighboring tribes, where they served particularly as medicine men, iron and copper workers, traders, and officials with ritual responsibilities. They traded throughout southern Africa. The Lemba have greater than 12 clans, some of which appear to correlate with place names in the Hadramaut. Parfit 1997. The Bubba clan is recognized as being the senior clan, both the oldest and, for some ritual purposes, the most important. Parfit 1997 has claimed to have discovered the original Sena of the Lemba in the eastern Hadramaut in the Yemen. Y-chromosome markers. The paternally inherited non-recombining portion of the Y chromosome includes polymorphisms that mutate relatively frequently, microsatellites, and biallelic polymorphisms, the YAP-ALU insert and single nucleotide substitutions that are unlikely to have arisen more than once in human evolution. For this reason, the latter group has been called unique event polymorphisms (UEP). Thomas et al. 1998, and it can usually be assumed to partition Y chromosomes into distinct genealogical groups. The ability to identify high-resolution haplotypes comprising linked markers mutating at different rates makes the Y chromosome a powerful instrument for investigation of relationships between geographically distant populations that may have been obscured through extensive admixture with their neighbors. Thomas et al. 1998. A previous study compared the frequency of RFLP polymorphisms P12F2, P49A, and PDP31 and the YAP insert in the Lemba and a number of other populations. Spertle and Jenkins 1996. The distribution of haplotypes identified was considered to be consistent with Lemba oral tradition but could not distinguish a Jewish from a more general Semitic contribution to the Lemba gene pool. Markers used in this study comprise 6 microsatellites: DYS19, DYS388, DYS390, DYS391, DYS392, and DYS393, and 6 UEP YAP, SRY4064, Psi81, SRY plus 465, 92R7, and TAT. The 12 polymorphisms listed above were characterized in multiple Jewish populations and identified a single haplotype, Cohen modal haplotype (CMH), that is dominant, frequency 0.50, in the Jewish priesthood and that may be more generally characteristic of Hebraic ancestry. Although the frequency is moderate, 0.12, in lay Jews, the CMH is absent or at low frequency in Yakut, Mongolians, Nepalese, Armenians, Greeks, and Cypriots. Authors unpublished data and, interestingly, in Palestinian Arabs. A. Nabel, D. Phelan, M. Feyerman, A. Oppenheim, Personal Communication. The combination of the presence of the CMH at high frequency in the Lemba and its absence in neighboring Bantu populations would be supportive of Lemba claims of a paternal Judaic ancestry, especially if its frequency is relatively low in other Semitic groups. Study populations. Lemba samples were collected from paternally unrelated but otherwise random males, identified as Lemba by the subjects themselves, on 2 separate occasions: in the Louis Tricart area of Northern Province, January 1997, 90 samples, and in Sekhukhune in Mpumalanga, October 1997, 46 samples, South Africa. Clan affiliations of 108.794 of the subjects were recorded. Bantu samples were collected February 1998 from paternally unrelated males of various chieftainships in the Pretoria area of South Africa who spoke a Bantu language. The subjects included 2 men born in Senegal who spoke Wolof, a non-Bantu language, as their first tongue. There is concern over the use of the word Bantu. Cavalli Sforza et al., 1994, p. 185. We follow Cavalli Sforza et al. in noting that, although Bantu was originally a linguistic term, its use to define population groups can be justified on the assumption that a geographic expansion spread both the Bantu language and a group of related people. Yemeni collections were made May 1997 at 2 locations in the Hadramaut, Yemen, from paternally unrelated but otherwise randomly selected males. Specific locations were the Sayan Teachers Training College in Sayan in the Hadramaut and Sena, a small population 3,000 isolated town located 60 kilometers east of Turam and 40 kilometers from the coast. The college draws its membership from a local but dispersed area and forms part of the new University of the Hadramaut. The present-day residents of Sena suggest that their ancestors may have moved to the area relatively recently, have a prolonged history of seafaring and trading with Eastern Africa and other overseas areas, including Southeast Asia, and has a considerable diaspora in Indonesia, Eastern Africa, Saudi Arabia, and Egypt. The people are Arab Muslims, although Jews are thought to have lived in the region during former times. Parfit 1997. Ashkenazic Israelites and Sephardic Israelites. Samples were collected from self-designated, paternally unrelated but otherwise randomly selected males in Canada, the United States, the United Kingdom, and Israel. In all cases, appropriate informed consent was obtained before samples were collected. Today, Jewish males can be divided into 3 castes: Kohanim, the paternally inherited priesthood; Levim, non-Cohen members of the paternally defined priestly tribe of Levi; and Israelites, all non-Cohen and non-Levite Jews. Significant differences in Y chromosome frequencies among these groups were recently reported by Thomas et al. 1998. As a consequence, frequencies of Y chromosome haplotypes of Jewish populations are expected to vary, in part because of differences in the proportion of Kohanim, Levim, and Israelites sampled. Since 90% of the Jewish population are Israelites, Bradman et al. In Press, it was decided, for the purpose of the current study, that, in terms of the data already available, Jewish populations are best represented by males of the Israelite caste. Data on some of the Ashkenazic and Sephardic Israelite samples have been the subject of publication elsewhere. Skorecki et al., 1997, Thomas et al., 1998. Population designations. Ashkenazic. These Jews follow the Ashkenazic rite associated with the Jewish communities of Northern Europe. Sephardic. These Jews follow a Sephardic rite associated with the Jewish communities of Northern Africa and Asia. The two communities have, to a great extent, been isolated from each other for 500 years. Unless otherwise noted, Semitic here will refer to the Jewish, Yemeni, and Sena populations together. Yemeni and Sena. Samples collected at the Sayan Teachers Training College are referred to here as Yemeni, samples collected at Sena as Sena. Sample collection and extraction of DNA in sample collection from Ashkenazic and Sephardic Jews. The subject lightly scraped the inside of the cheek with a wooden spatula, swished saline solution around the mouth for 30s, and expelled the solution into a collection tube. Buccal swabs wiped over the inside of the cheek and returned to collection tubes to which 0.05 MEDTA, 0.5% SDS was added. DNA was extracted by use of a standard phenol procedure. Typing 2 multiplex kits, one for the 6 microsatellites and the other for the 6 UEP, were developed for use with an ABI 310 genetic analyzer. Thomas et al. 1998. 5% of the samples were retyped for microsatellite repeats on an ABI 377 genetic analyzer in a separate laboratory. Only 1/120th typings were different, DYS 392, a difference of 2 steps. Genetic distances, haplotypes, and genealogical trees. Genetic distances for haplotypes were calculated with use of the computer program Microsat to infer genealogical trees that were drawn with the program Philip Felsenstein 1993. Genetic similarities of populations were calculated by use of Nay's 1987 genetic identity. Microsatellite haplotypes present at a frequency 0.1 in any of the 6 populations were included in a table of frequently encountered haplotypes. Results. UEP groups. Table 1 classifies the Y chromosomes of the 6 sample populations into 4 groups, for a cladogram illustrating the evolutionary relationships of the 4 UEP groups. See Fig. 1. The distribution of the 4 UEP groups among the populations is apparently consistent with earlier suggestions that the Lemba have a mixed Bantu Semitic origin. The frequency of the YAP plus chromosomes in the Lemba is intermediate between that of the Bantu group and that of the Semitic group. However, the distribution between the 2 YAP plus groups is significantly different. Only 2 Bantu, 4 Lemba, and 3 Yemeni Y chromosomes are included in UEP Group 3, YAP plus Psi 81A, whereas all of the YAP plus Israelites fall into this group. YAP plus Psi 81A Y chromosomes have been identified in many widely separated populations in Asia, Africa, and Europe. It is more widespread than the derived YAP plus SY81G haplotype, UEP Group 4, which appears to be substantially confined to Africa. Hammer et al., 1997. Given the significant difference between the distribution of the 2 YAP plus UEP groups in the Semitic and Bantu populations, interpreting the total frequency of YAP plus Y chromosomes as an indicator of admixture in the Lemba could be misleading. Distinguishing the frequencies of the YAP plus Psi 81A and YAP plus Psi 81G Y chromosomes, however, seems to provide a more appropriate level of resolution. All members of the Sena population fall within UEP Group 1, the most common UEP group, YAP-92R7C, in the Semitic populations. The relatively greater homogeneity of the Sina population may result from the town's isolation and from its catchment area being more limited than those for the other Hadramaut samples. Although the Ashkenazic Israelites, Sephardic Israelites, and Yemeni are not significantly different from each other in the distribution of Y chromosomes across UEP groups, all other populations are both significantly different from one another and from the Ashkenazic Israelites, Sephardic Israelites, and Yemeni Paul cases. Genetic identity. Ney's genetic identity I, calculated on the basis of distributions of haplotypes at the UEP level, shows the Lemba to be closer to the 4 Semitic populations than to the Bantu. Haplotypes. Table 2 lists the Y chromosome haplotypes of all samples, classified by UEP group and microsatellites, identifying those Y chromosome haplotypes that are observed in more than one population. Only 2.014 of the 142 microsatellite haplotypes are duplicated across UEP groups, which provides some confidence that the 6 microsatellites used in this study are sufficient to prevent high levels of homoplasy, at least at fairly deep genealogical levels. Evolutionary convergence of the microsatellite haplotypes within UEP haplogroups will, of course, not be detected. To evaluate sharing of Y chromosomes between populations, we considered both the total number of Y chromosomes observed Table 3 top and a data set in which each Y chromosome haplotype was counted only once Table 3 bottom. The degree of sharing of Y chromosomes between Ashkenazic and Sephardic Israelites has been reported elsewhere, Bradman et al., in Press. Haplotype 34 CMH is present in varying frequencies in Ashkenazic Israelites, Sephardic Israelites, Yemeni, and Lemba but is absent from both the Bantu population and the Sina population. The CMH represents 0.088 of all Lemba Y chromosomes and 0.135 of Lemba UEP Group 1 Y chromosomes. Equivalent population, UEP Group 1, proportions for Ashkenazic Israelites, Sephardic Israelites, and Yemeni are, respectively, 0.150, 0.231, 0.100, 0.161, and 0.020, 0.027. The CMH is an important component in the sharing of Ashkenazic and Sephardic Israelite Y chromosomes. It is also the major component of Ashkenazic and Sephardic Israelite Y chromosome sharing with the Lemba. The Yemeni Hadramaut samples only have one such chromosome among them. Two striking features are the large proportion of haplotypes found in the Bantu that are also present in the Lemba 0.448, Table 3, bottom, and the even higher proportion of Y chromosomes 0.740, Table 3, top. Gene diversity. Table 4 gives gene diversity, Ney 1987, for all 6 populations, at the UEP group level and at the 12-marker haplotype level. The gene diversity is calculated separately for each UEP group, for all populations having 10 Y chromosomes within that group. Geographic isolation may be the reason why the Sina population is the most homogeneous at the all haplotypes level. Among the other 5 populations, the homogeneity of the Bantu is striking, particularly at the UEP group level. Haplotype 117 and its one-step microsatellite neighbors comprise almost half the Bantu Y chromosomes and could be a candidate signature haplotype of the Eastern Bantu expansion. It will be interesting to see whether the haplotype cluster is represented in other Eastern Bantu, as well as in Western Bantu and Cameroon Bantu populations. The 2 Wolof individuals shared the same haplotype, haplotype 128, a one-step neighbor of the Bantu modal haplotype, with each other and with 2 Pedi individuals, 1 Swazi individual, and 1 Ndebele individual. The Bantu modal haplotype was not significantly associated, p equals 0.773, with any single tribal group, Raymond and Rousset 1995. Furthermore, no particular association of tribe and haplotype was immediately apparent. Approximately 78% (60/77) of Y chromosomes were shared across tribes, whereas 45% (13/29) of Y chromosome haplotypes were similarly shared. And 39 together represent more than 1/3, 40%, n equals 54, they represent greater than 60% of that group. Microsatellite variability. As reported by Thomas et al., 1998, the distribution of alleles across UEP groups suggests that microsatellite DYS388 does not mutate in a consistent stepwise manner over its entire range of repeat counts. The distribution of DYS388 alleles in UEP Group 1 is strikingly different from that in other UEP groups. UEP Group 1 has a two-peak distribution in which most alleles are either 12 or 15, whereas in other UEP groups in this study its repeat length is never greater than 13. It is possible that low and high repeat number subgroups within UEP Group 1 are genealogically distinct, but with current data, it is not possible to exclude the possibility that frequent large deletions occur in high repeat number alleles, as has been observed in artificially constructed systems. Weirdle et al., 1997. Kaiser et al., 1997, analyzing European samples, reported a lack of variation at DYS388, with all samples typed having a low repeat number allele. All Bantu samples in the present study also had low repeat number DYS388 alleles, whereas Semitic populations have a majority of Y chromosomes with high repeat number DYS388 alleles. The typing of additional populations may reveal whether high repeat number DYS388 alleles common in Near Eastern populations are found at high frequency elsewhere. For now, high repeat number DYS388 alleles appear to be diagnostic of the Near East. The distributions of alleles of the other 5 microsatellites are similar to the distributions reported by Kaiser et al., 1997. Frequently encountered haplotypes. 9 haplotypes are represented by Y chromosomes present at a frequency 10% in one or more of the 6 populations. Figure 2 shows the populations in which they are found. The figure clearly illustrates the relationships uncovered in tables 11 and 3, namely, the apparent contributions of Bantu and Jews to the Lemba Y chromosomes, as well as the connection between the Yemeni and the Sena populations. It also highlights haplotype 39, which is private to the Lemba and constitutes greater than 11% of the total Lemba Y chromosomes and 18% of the Lemba Y chromosomes in UEP Group 1. The origins of Lemba Y chromosomes. To explore with greater resolution the possible origins of Lemba Y chromosomes, genealogical trees were drawn that were based on microsatellite variation. These were used to assess, in the case of each Lemba haplotype, whether each has a close genealogical relationship with one or more haplotypes present in any of the other 5 populations. Separate genealogical trees of the individual haplotypes were drawn for each UEP group by use of 2 distance measures, average squared distance (ASD) and proportion of shared alleles, Slatkin 1995, Goldstein and Pollack 1997. ASD trees for UEP Group 1 haplotypes were drawn, with and without DYS388. As expected, given the high versus low repeat number allele distribution, inclusion of DYS388 increased the internal structure of the UEP Group 1 tree, with haplotypes containing high repeat number DYS388 alleles grouping within one part of the tree and with haplotypes with low repeat number DYS388 alleles grouping within the other part of the tree. Fig. 3. The tree also shows the populations in which each haplotype is represented. As would be anticipated with a large sample of similar haplotypes, bootstrap values were usually low, with only 3/77 nodes recording values greater than 60% after 500 resamplings. Classification into UEP group and position in genealogical trees differentiated Semitic and Bantu haplotypes. The data in Table 3 demonstrate that there is very little sharing of Bantu and Semitic Y chromosomes. The highest level, at 9%, is due to a single Bantu haplotype shared with the Yemeni population. Consistent with a deep genealogical division between the 2 groups, most Semitic and Bantu haplotypes could be clearly differentiated either by their membership in a UEP group associated with Semitic or Bantu haplotypes or by their positions in a microsatellite-based tree restricted to a single UEP group. In UEP Group 1, for example, the Bantu Y chromosomes clustered within their own clade. Lemba Y chromosomes, on the other hand, were frequently closely associated with either Bantu or Semitic Y chromosomes but only very rarely with both of them simultaneously. Given that differences can be observed between Semitic and Bantu Y chromosomes, it is possible to suggest a Semitic or Bantu origin for Lemba Y chromosomes, on the basis of their genealogical proximity to Bantu and Semitic types. In UEP Group 1, the two Lemba Y chromosomes, haplotypes 52 and 85, that are members of an otherwise exclusively Bantu clade are classified as Bantu. Haplotype 57 Y chromosomes are classified as Semitic, since they are one-step neighbors of a Sephardic Israelite and a Yemeni, whereas the nearest member of the Bantu clade has an ASD distance 6 times greater. All other Lemba Y chromosomes in this haplogroup are classified as Semitic, since they are included within Semitic clades in the genealogical tree. Since all other Y chromosomes in UEP Group 2 are from Semitic samples, the two Lemba Y chromosomes are classified as Semitic. The 4 Lemba Y chromosomes in UEP Group 3 belong to a haplotype shared with 3 Israelites and are, therefore, classified as Semitic. Only 2 1/5 Y chromosomes in UEP Group 4 are Semitic, both are Yemeni, and one of these two is shared with the Lemba and Bantu. Of the Bantu Y chromosomes not included within the Bantu clade in the UEP Group 1 tree, 96.9% are included in UEP Group 4. For these reasons, the Lemba Y chromosomes in UEP Group 4 have been assigned a Bantu origin. The consequence of the above assignments is to designate 92 (67.6%) Lemba Y chromosomes as having a Semitic origin and to designate the other 44 (32.4%) of them as having a Bantu origin. The Lemba clans. For 108 of the 136 Lemba males sampled, clan affiliation was ascertained by self-identification. More than 10 samples per clan were collected from members of 6 clans: Bubba, Haji, Hamasi, Mani, Siddiqui, and Thuhakale. All 6 clans were represented in both the Northern Province and Sekakunland collections. Only the Mani clan showed a significantly different distribution. P clans contained both UEP Group 1 and UEP Group 4 Y chromosomes, but none displayed UEP Group 3 Y chromosomes. The Y chromosome distribution among UEP groups was not significantly different, p greater than 0.34, between the two collection places. Of particular interest is the Bubba clan, since membership of this clan and possession of the CMH are significantly associated. Y chromosomes came from members of this clan, whereas 7 Northern Province, 4 quarters, and Sekakunland 3/9 of the 13 Bubba have the CMH. F.C. Ralinga Hamasi, a Lemba elder, in a speech at the burial of Manda William Mawela Ratchilingana Mani, in July 1996 before the current research was undertaken, said that the Sinas left Judea under the leadership of Bubba and settled in Yemen where they built their city of Sena, hence Sinas, reflecting the belief of at least one elder that Bubba led the Lemba out of Judea. On the other hand, the Encyclopedia Judaica 1972 makes no mention of a Bubba in Jewish history. In a book published privately in 1992, another Lemba elder wrote that the Buba lineage came down from Judea as the leading lineage of the Basina when they left Judea in their early migration to the Yemen where they settled and built the city of Sena. They ruled over all the lineages in good manner. Mathivha 1992, p. 23. The Bantu expansion Bantu Y chromosomes are found in UEP Group 1, UEP Group 3, and, most frequently, in UEP Group 4. The proportion of YAP-Bantu Y chromosomes found in this study is consistent with the results of other investigators, who report a significant YAP- element in the Eastern Bantu. Hammer et al. 1998. The 7 Bantu haplotypes, one of which is represented in the Lemba, and the Lemba haplotype 85 formed a distinct clade in trees based on use of both ASD and allele sharing as distance measures. Bootstrap support 45% in 500 resamplings of the ASD-based tree is moderately good, given the large number of haplotypes in the tree and their generally close inferred genealogical relationships. It may be that the 8 Y-chromosome haplotypes in this clade are representatives of a YAP- genealogy present in the eastern Bantu, alongside the dominant YAP plus Y chromosomes. It will be interesting to see whether these or similar haplotypes are represented in other Bantu populations. There is considerable archaeological and linguistic evidence to support an expansion of Bantu-speaking people throughout subequatorial Africa. Cavalli Sforza et al., 1994. It is therefore interesting to see what dates are suggested by the level of microsatellite variance among the dominant, UEP group 4 Bantu Y chromosomes typed in this study. When there is good reason to believe that the correct ancestral Y chromosome can be identified, for example, by its presence at a high frequency, both alone and together with its one-step neighbors, it is possible to use the method of Thomas et al., 1998, in which the time to coalescence to an ancestral Y chromosome is ASD equals μt. A single-step mutation model is assumed, with mu being the average microsatellite mutation rate per generation and with t being the time in generations. Haplotype 117, which is present across the Bantu tribes and in the Lemba, was selected as the ancestral Y chromosome. Alone it represents 25.8% of UEP group 4 Bantu sample Y chromosomes, 58% together with its one-step neighbors. When the ASD for microsatellites DYS19, DYS390, DYS391, DYS392, and DYS393 is calculated and mu equals 0.0021, Higher et al., 1997, and 25 years generation are used, the time to coalescence is 4,839 years. We also used the same approach as did Kittles et al. Slack in 1995, Kittles et al. 1998, to calculate an approximate time for the start of rapid population growth. In this method, V equals μt, where V is the average variance of microsatellite repeat counts. Using the same microsatellites, mutation rate, and generation time as used in the ASD equals μt calculation above produced a time of 3,310 years. Both times accord with estimates of the agricultural and Bantu expansions in subequatorial Africa, i.e., 3,000 to 5,000 years before the present. Cavalli Sforza et al., 1994. It should be appreciated that these dates carry very large confidence intervals, because of variability and uncertainty associated with 1. The evolutionary process and 2. The mutation rate and process. Since the estimates of confidence intervals depend on assumptions about demographic history, Goldstein et al., 1999, we have not attempted to estimate them here.